This diagram is going to help us understand the different types of membrane transport. So again, membrane transport is how molecules gain access to the inside of a cell or how molecules are moved outside of a cell through the plasma membrane. We can take that idea and divide it into two methods. One method is called active transport. The other methods are called passive transport. Active requires ATP. Passive requires no ATP. So you'll notice all of these mechanisms over here require energy for them to work. These mechanisms here, simple diffusion and facilitated diffusion, do not require energy. So why? Why would one mechanism need energy and the other would not? Well, it has to do with concentration differences. If you are trying to move a molecule from an area of low concentration to an area of high concentration, it requires energy. If you are moving a molecule from high concentration to low concentration, it does not require energy. And we often call that diffusion. So I'll repeat, if it's low to high, it's active transport, requires energy. If it's high to low, you do not need energy. So let's look at those non-energy required methods first. Simple diffusion. Simple diffusion is when molecules move down their concentration gradient, that means high to low, but they can diffuse right through the membrane with no need for a transport protein. So that's the key difference between simple and facilitated. If you look at that word facilitated, when you facilitate something, you help it. Facilitated diffusion is when the molecule needs help going through the membrane. What helps that diffusion? Carrier proteins, channel proteins, gated channels, leakage channels, or aquaporins. We're not going to get into too much detail now, but we will in the future. But what these all have in common is there needs to be some sort of a protein in the membrane that helps move that molecule from an area of high concentration to an area of low concentration. If we look at the active transport side, one thing is you're moving molecules against their concentration gradient. That means you are moving them from an area in that they're low concentration to an area of high concentration. It's like moving something uphill. You need more energy to move things uphill than letting things go downhill. We often call these active transporters. They will either require a vesicle or require a membrane protein. If you require a vesicle, we call you vesicular transport. You can move things into a cell, that's called endocytosis. You can move things out of a cell, called exocytosis. You can move things through a cell, that's called transcytosis. So transcytosis is really the combination of endocytosis on one side of a cell, exocytosis on another side of the cell. And if you think about that, that in essence is moving that molecule right through the cell because it enters and then exits. Vesicular trafficking is just a way we can move molecules around within a cell. All these mechanisms require energy. And they require a vesicle. A vesicle is a little container, a membrane enclosed container. Primary active transport and secondary active transport are through a membrane, require a protein transporter, but it's going against its gradient. We're not going to talk too much about secondary active transport, but secondary active transport depends on first establishing an ion gradient, then moving a different ion. The example is if we move sodium into a cell, that's called primary active transport. What that does is you're moving a positive ion in one direction. The secondary active transport would be moving the negative ion towards that positive sodium. So in order to move the negative ion, like chloride, you first had to move that positive sodium ion. So that's called primary and then secondary transport. Okay, next thing are these tables that will go over this information. Kind of again, a little more text here that you can use to study. But these are the passive processes we just mentioned. Diffusion, which can be divided into simple diffusion, facilitated diffusion, or osmosis. Osmosis is just diffusion of water. So you can read through these definitions on your own. But notice this drawing. There is the drawing for simple diffusion. Okay, so we're moving this molecule right through the membrane. This molecule is in a high concentration on this side, a low concentration on this side. So it moves through the membrane without the need of a transporter. These two mechanisms are both facilitated transport. These molecules are moving from high concentration to low concentration, but they require some sort of a membrane protein, whether it be a channel or a carrier. If we look down here on these drawings, this shows osmosis. Osmosis is when you have a concentration difference of water. So if we look on this side of the tube compared to this side of the tube, there are more water molecules here. Why is that? That's because there's solute molecules over here. A solute are other molecules in that water. So water will want to move from this side to this side, from the left to the right, because there's less water here, because there's solute molecules. So now we see that water moving from left to right. We call that osmotic pressure. Osmotic pressure is the pressure due to that movement of water. So you could actually measure pressure against this component. And that pressure is called osmotic pressure. So let's review what water does. Water moves from where water is in high concentration to where water is in low concentration. Said slightly differently, water moves towards high solutes. Notice there's a lot of solutes here. Let's say those solutes are sodium chloride. If you've got salt water here, pure water over here, water is going to move towards that salt water. Why? Because there's low water and high solute concentration. So those are some examples of passive transport to be familiar with. This shows how important understanding this information is. When we give someone fluids into their bloodstream, that fluid in those IV bags has to have a certain amount of solute and water in it. When we put a cell in a solution that has the same concentration of solutes, we call that an isotonic solution. Iso means same. So this fluid out here has the same concentration of solutes as the inside of this red blood cell. What happens is water moves in and out of that cell, but equally, and that cell maintains its shape. If you put a red blood cell in a hypotonic solution, hypo means low. What it's referring to is low solute concentration. So this would be like putting red blood cells in pure water. Pure water is as hypotonic as it gets because there's nothing else in that pure water. that means that the concentration of water here is much higher than the concentration of water in the red blood cell. So water will tend to move into the red blood cell, and that cell will swell and possibly rupture. We call that hemolysis. So when you put red blood cells in hypotonic solutions, they swell and burst. If you put a red blood cell in a hypertonic solution, hyper means that there's a lot of solutes out here. Water will move towards that high solute, and the cell will shrivel up, and we call that crenation. Both of these situations in panel B and C are not very healthy for this red blood cell. This cell ruptured and this cell shriveled up. So IV bags that we give people, maybe that IV bag contains an antibiotic or some other medicine, and we're putting fluid directly into the bloodstream, that fluid has to be isotonic to our blood cells. You never put pure water in someone's bloodstream. You never put high salt water in someone's bloodstream. So these are important topics to understand when you move on to, say, nursing. Here's a review of active transport. It's just a table. You can read through it on your own. I would definitely study and understand the basic idea here for all these mechanisms is they require energy to move that molecule against their concentration gradient. That's another way of saying moving from low to high concentration. It takes energy to do that. The next video we're going to start here and learn about the most important active transporter called the sodium-potassium